Welcome to the DiabetesBio podcast. This is a new podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. Each month, we will interview one or more authors from the feature articles, the articles of the month, or articles published in that month's issue of the Diabetes Journal that we find interesting. We also have a Sweet Talk segment of the podcast, which is more of an open format discussion of additional articles, conferences, or major events in the field that we'd like to highlight. My name is Darleen Sandoval, and I am joined by my podcast co-hosts, Dr. Kirk Haberger and Dr. Kevin Williams. Good to see you both today.
Hey, Dar, good to be here.
Hello, hello.
So I don't know about you guys, but we're having a beautiful day here in Denver, Colorado, getting a little break from the winter grays and snow.
Yeah. Actually, here in Birmingham, it's blue skies, and feels like spring might be close. It's got to be about the same for Kevin.
I know the listeners can't see it, but behind me you can see the blue skies. That's not a painting, so it's quite beautiful right now.
Yeah. It's about time.
All right. Kirk, why don't you tell us about the articles we'll be featuring this month?
Absolutely. So, Dar, before I get started, I want to make a quick comment on the paper of the month. The title of that paper is High Doses of Exogenous Glucagon Stimulate Insulin Secretion and Reduce Insulin Clearance in Healthy Humans. Now, this is work from Dave D'Alessio's group, including collaborations with John Campbell. And we've actually just heard from both Dave and John in recent episodes. So we thought that we might use this episode to highlight other investigators' work that will be in this month's edition of the journal. John also gave us a little bit of a sneak peek on that paper in last month's Sweet Talk. So again, just a chance to sort of broaden our horizons, but definitely check out that paper. It's excellent work, and all things glucagon are good, right? All right, so now on to our two articles. The first is Dual Tracer Test to Measure Tissue-Specific Insulin Action in Individual Mice Identifies in Vivo Insulin Resistance Without Fasting Hyperinsulinemia. This is work that comes to us from David James and his group and is led by a young scientist, Harry Cutler. Really interesting stuff. I'm looking forward to this conversation. The second is work from Lori Sussel, who's from your neck of the woods there, Darleen.
That's right.
And she is going to join us to talk about her work on a protein tyrosine phosphatase N2 and its regulation of metabolic flux, which appears to affect beta cell susceptibility to inflammatory stress. So good papers here this month.
This month, we have an extra special, separate bonus episode of our Sweet Talk segment. So Kevin is going to record a whole other episode of our podcast. Kevin, tell us what's happening today.
That's right. We will be highlighting a brand new set of feature articles in the journal that focus on the work and careers of the Pathway to Stop Diabetes Award recipients. Many of you listening are probably aware, but some of you may not be, that the Pathway to Stop Diabetes Award initiative was launched by the American Diabetes Association about 10 years ago. This award focuses on innovative and transformational approaches to prevent, manage, and cure diabetes. We've got a two-part segment lined up. First up, we will be talking to Dr. Christopher Newgard from Duke University and Dr. Lou Phillipson from the University of Chicago. They are both co-chairs on the ADA Pathways Mentor Advisory Group, or MAG, and they will be filling us in on what this program's all about and what's on the horizon. Then in part two, we're going to catch up with Dr. Celine Riera from Cedars-Sinai in LA. Dr. Riera is a recipient of the Pathways Initiator Award and kicks off our article series with a piece she wrote for the March 2024 edition of the journal titled Wiring the Brain for Wellness, Sensory Integration in Feeding and Thermogenesis. So I really encourage our listeners to check out this special podcast segment, as I think it's going to be a fascinating topic. I know I'm really looking forward to hearing more about this program and where it's headed.
Okay, and now on to today's interviews.
Our first feature article from the March 2024 issue of Diabetes is titled Dual Tracer Test to Measure Tissue-Specific Insulin Action in Individual Mice Identifies in Vivo Insulin Resistance Without Fasting Hyperinsulinemia. We are joined by two authors of this paper, Harry Cutler and David James. Mr. Cutler is a PhD candidate in the School of Life and Environmental Sciences, and Dr. James is the Leonard P. Ullman Chair of Metabolic Systems Biology and ARC Laureate Fellow at the Charles Perkins Center of the University of Sydney. Welcome, Mr. Cutler and Dr. James. Congratulations on your paper, and thank you for being part of this month's podcast.
Nice to be here.
Thanks, it's a pleasure to be here.
So we like to start the podcast with a fun question, and we're going to actually focus on a U.S. Twitter holiday. March 14th is National Scientist Day, and that actually coincides with Albert Einstein's birthday. But to commemorate this day, can each of you tell me if you have a favorite scientist, either current or historical, that you find particularly inspiring? And Harry, you can't choose your mentor, so but that gets you off the hook there. Why don't you start, Dr. James?
Okay, so there are many people that have inspired me throughout my career. It's probably because it's a long career. But one of those who was particularly inspiring for me was George Palade. George Palade, I'm sure, is known to all of us as one of the sort of fathers of cell biology. He pioneered the use of the electron microscope to study how molecules move around inside the cell. And I was privileged to meet him in the late 1980s when I was a postdoctoral fellow at Washington University in St. Louis.
Nice.
We had just discovered GLUT4, and I met with George and I told him all about GLUT4, and he just sat there very pensively listening. And then at the end of the conversation, he turned to me and he said, "I believe you're dealing with a case of regulated exocytosis." That's what he said. And he was a very inspiring man, very inspiring. So that would be my choice for today.
That's a great one. Mr. Cutler, do you have one in mind?
Yeah, I sort of grew up as a kid, always wondering how things worked and why the universe was the way it was. And one of my inspirations was actually a TV presenter called Brian Cox. And he had this TV series where he would explain sort of how the universe works and how stars were born. And that's very different from my current interest, but it sort of sparked a lifelong curiosity for finding how things worked, and especially mechanisms.
Yeah, that's awesome. The U.S. equivalent, I guess, would be Bill Nye the Science Guy.
And
Yeah.
no, that's the first one that came to mind for me too. So yeah.
Awesome. Kirk?
All right. So Dr. James, can you briefly summarize your research program and maybe a bit about what led to this specific project?
Yeah, that would be my pleasure. My lab and my interest for a long time now has centered around the governance of insulin over metabolic homeostasis. And of course, in that interest, one can't help but also be interested in the mechanisms that lead to insulin resistance. And the reason for that is that, I still believe that insulin resistance is a major path to many, many diseases. And if we could figure out how to solve insulin resistance, the belief is that you could, in one fell swoop, eliminate a whole swath of diseases, which I think is, big. Now, how did we get into this project? So our work is really, we do work on humans as well, but we do a lot of work in mice. There are two things that led to this project, really. One was our desire to measure tissue-specific insulin action. Insulin, as you know, works at a whole body level, mainly in muscle, adipose tissue, and the liver. And so really, I think it's critical in trying to understand how insulin works to sample insulin action in all of those tissues, preferably simultaneously in the one system. So that was number one. Number two is very recently, we have become extremely interested in the impact of genetics on metabolism. Like so many others in the field, we used to work in the C57 Black 6 mouse. Our work was confined to that little guy. And, one day I realized how silly this was in a way, because it's a little bit like working in one human being over and over and over and over again with the idea that, you're going to understand how stuff works, right? And I think that's a bit limited. So we started working on many, many different genetic backgrounds of mice. And so again, what we realized is we needed methods, not just to sample tissue-specific insulin action, but insulin action across many, many different mouse strains. And that really provided us with a challenge and set up this particular study that we're very proud of.
Yeah, very, very good. Now, I want to switch to Mr. Cutler here. And can you maybe build off of that and tell me about your research interests and, how you plug into what you're thinking about doing after your PhD?
Yeah, sure. So as David was kind of alluding to there, we've taken this technique that we've developed in this paper and now applied it to many different inbred strains. And some of these are classic lab-derived strains like the Black 6 and the DBA and 129s, but others are quite new wild-derived strains like cast mice and WSBs. And these are some of the founders for the wider genetic reference panels like the diversity outbred mice. And what we found in these animals is that insulin resistance occurs quite specifically in individual tissues. And it's not that insulin resistance occurs across all tissues at once. And so we think that insulin resistance really is a tissue-specific phenomena in these mice. And the other thing that's of interest here is that these individuals from the different strains display very different disease susceptibilities. And so some of them develop disease quite quickly when you feed them a high-fat diet, whereas others are very resistant to that. And presumably that also goes on in the human population, where we see that some people are very susceptible to developing type 2 diabetes. And so my research interests at the moment are really in understanding the molecular mechanisms of that disease susceptibility. I think the question as to why some individuals don't develop disease is just as interesting as why some individuals do. And I think the former of those hasn't really been tackled by the research community more generally. So we've been using genetics, but also techniques like mass spectrometry-based proteomics to answer this. And so I think continuing on, I'd be really keen to pursue those sorts of directions.
Great. Dar?
All right. Dr. James, you touched on this a little bit, but in this manuscript, you describe a dual tracer test that allows for assessment of both basal and insulin-stimulated glucose uptake. So from a broad perspective, can you tell us why this endpoint is important for the field?
Yeah, thank you. Let me just go back one step, if I may, and just talk about the method in itself. So, as I said before, we were particularly interested in a method that could assess tissue-specific insulin action all in the one setting. And, if you look at the methods that were available up until this point, we had things like the glucose tolerance test, which I think is the most commonly used method. Everybody knows about the GTT. It's very easy to do. Many labs do it. But the problem is it's not really clear what it's measuring. And I think the reality is it's probably measuring many, many different things. And so that has led to, I think, a little bit of, a few misconceptions in the field. So that was problematic. Now, the best method, I think, without question, is the hyperinsulinemic euglycemic clamp, because, it's a very well-controlled scenario where everything is at steady state and so on and so forth. And if you combine that with traces, you can get really beautiful insights. But the reality is that really, with the exception of a handful of labs, like David Wasserman and at Vanderbilt and Jason Kim at UMass and others, with the exception of them, it's a really hard method to do. It's not something that the average lab can just start doing. And that's a problem. So now the other issue, which is key to this method, we call it the DTT, by the way, the dual tracer test, playing on the GTT. Harry came up with that. And the key is that, many methods only assess insulin action without looking at what happens under basal conditions. And that's problematic because if you see changes in the baseline or the ground state, you may think that there are changes in how insulin works, but it's not until you can see that relative difference between the baseline and insulin that you're truly able to judge insulin action. That really is the definition of insulin action. And, if you have different genetic models, it's really hard to do baseline measures, then insulin measures separately in the one animal. You really want to be able to do it together. And that is the great strength of our method, A, because you can measure both baseline and insulin together, but B, we believe our method should be applicable to any lab in the world. If you have very fundamental mouse skills, our method, I think, should be available to the average metabolic scientist. And I think that's a real strength.
Absolutely. I just want to commend Harry for not naming this the Cutler protocol, right? Because that would have been my go-to. So, hats off.
It's got a kind of rig to it, the Cutler protocol.
I'm just saying.
Yeah, maybe the next one.
So I guess, Harry, moving to you, tell me more about the basic methodology and the concepts behind the dual tracer method. And I'm curious specifically if there's a reason why you've used the C13 for the basal and the tritium label for the insulin-stimulated glucose labeling.
So you raise a great point there that the basic kind of unit of this test is in the tracers. And so in order to have one for basal uptake and one for insulin-stimulated, they need to be differently labeled because you need to be able to separate them from the same tissue at the end. Now, it doesn't matter which way you put it, in theory, because both of those tracers, as far as we can tell, are taken into the tissue at exactly the same affinity. And so the only reason that we've chosen to put the tritium second is because that has a much lower counting efficiency than the 14C. And so what that means is that you need to put about 3 times more tritium into the beta counter to get the same sort of efficiency and accuracy as you do for the 14C. Now, if you put the 3H first, so more of it as the basal tracer, that would mean that you were then adding lots of basal tracer that would then be more present when you give insulin. And so we reasoned that we would reduce the sort of artifactual uptake of that basal tracer during the insulin-stimulated phase if we gave the tritium second. The other sort of fundamental aspects of this assay are that we really care about muscle glucose uptake. And in mice, which aren't like humans, which just sit on a bed while you measure their insulin action, mice, as soon as you inject them with anything, will run around the cage like crazy. And what that means in a mouse where exercise is 10 times more likely, better able to stimulate glucose uptake than insulin, is that if you don't control for activity, then that's a massive confounder for your ability to measure insulin sensitivity. And so for that reason, we've anesthetized the mice, and we get really clean data from all those muscles. It's very consistent. And so we've been quite happy with that compromise. And as David sort of was talking about, with this being a simple assay, I'd just like to iterate that I was actually an honors student when I developed this assay. So it was my first week in the lab, hadn't ever touched a mouse before. And yeah, this was my first project.
Great job. Can you, this is a follow-up on that question about the anesthesiology. So which anesthetic did you use? Because those, the various different types of anesthetics can in and of itself affect glucose uptake.
Absolutely. So isoflurane is the most notorious of those that will put blood glucose through the roof instantly. We use pentobarbital in this instance, and we found that the increase in blood glucose is not really that much more pronounced than just injecting saline. And so we think that the increase in blood glucose is largely just a stress response.
Nice. One of the unique aspects of your paper, at least one aspect that I really appreciated, was that you really did a variety of validation experiments for this technique. So Dr. Cutler, can you highlight some of those key findings and those key results that really led you to believe this would be a valid approach to studying glucose uptake?
Sure. So our first question was, how independently can you separate out basal and insulin stimulated? Because if you're making a point that you can measure basal independently, I think the onus is sort of on us to prove that and to convince people that that's what we're doing. And so one of the first experiments that we did was to put a basal tracer into an animal and then either leave it for a full 80 minutes or at 40 minutes, give it insulin as well. And then we compared how much of that basal tracer is in all of the tissues. And what we found is that for most of the tissues, excluding heart, the level of the basal tracer that accumulates is no different regardless of whether it's seen insulin or not. And so that really convinced us that we could measure basal glucose uptake independently of insulin action. And then once we had sort of convinced ourselves of that, we wanted to look at whether we could find similar results in a diet-induced model of insulin resistance as you would by claim. And we wanted to compare it to the hyperinsulinemic glycemic clamp because that's such a gold standard method. And we found all of the insulin resistance that you would expect. We found it in white adipose and skeletal muscle and so forth. And then finally, we wanted to see in a genetic model, what would insulin resistance look like in the model of GLUT4 deficiency? And GLUT4 obviously is such a key protein in insulin action. And so the expected results there are very clear. And we think that by having these multiple validation steps, it's really convinced us that we have a lot of faith in our results.
Excellent.
Great. So, Harry, another question we landed on in reading your paper was your assessment of the variation both between mice and even within samples, or within each mouse. Can you tell us a little bit about those findings and then their implications for the field?
Yeah, I think all too often the variation in inbred strains is something that gets ignored and sort of swept under the rug a little bit. And so an example of that is what happens to the fat mass of let's just take a C57 black 6. And on chow, let's say that the fat mass of that mouse is going to range between maybe 5 to 10 grams in an adult mouse. When you feed it a high-fat diet, what happens is that the range of fat masses in that population will go from 5, as it was, to maybe 30 grams. And so even though these mice are all genetically identical, some of them will remain at 5 grams of fat. And so the variation increases massively, but you need to account for that when you're measuring any sort of metabolic phenotype. And so if in the case of many single tracer tests where they measure basal and insulin-stimulated glucose uptake in separate animals, what you're doing there is sort of comparing apples with oranges because the insulin-stimulated mice are not the same as the basal mice. They're different mice. But if you can measure both of those glucose uptakes in the same animal, now you're comparing apples with apples.
Yeah, great point.
All right, Dr. James, can you finish off our discussion by highlighting, basically summarizing the strengths and also the limitations associated with the approach, but then giving you the opportunity to provide that sort of take-home message of why this is an approach that we should be utilizing as the field?
Yes. So the strengths, as I already alluded to, are first simplicity. I think the method is relatively simple and should be able to be adopted in any lab, and I think that's a real positive. Second, it's very sensitive for being able to measure particularly muscle insulin action. And you know what, in our experience, this is not something that is easy to measure in a mouse. It's actually very difficult, for example, to pick up insulin resistance, let's say in a high-fat fed mouse in skeletal muscle. And if you look through the literature, there's a lot of variance in people's ability to report this. Whereas in this model, we were able to see it and, quite consistently. So I think that's also a great strength. Now, as is the case for any model system in science, it also has its weaknesses. One is we are studying these phenomena under non-steady state conditions because we give a dose of insulin and the blood glucose is falling as we are assessing insulin action. And, that's not without its limitations because, you're not measuring this under steady state. Second, as already discussed, is the use of anesthesia. Again, we do believe that, if you look at other models, for example, a lot of people look at insulin action ex vivo in bits of muscle in test tubes. I think this method, despite the anesthesia, is superior to that. And then thirdly, I would say our method is not amenable or not easily amenable to assessing the liver. And that's a great pity because, as we all know, the liver is a major target. And 2-deoxyglucose is, many people may not realize this. It is not useful for looking at metabolism in the liver because the whole premise of its use is that when it goes in, for example, into a muscle cell, it's phosphorylated by hexokinase. But those cells do not express phosphatases that can dephosphorylate the tracer so it could then move in and out at the same time. So in muscle, once it goes in, it stays in and accumulates there. And that's a big strength. In the liver, that's not the case. You cannot use it in the liver. So I think they're the main strengths and weaknesses.
Awesome. And any take-home messages?
There's so many take-home messages. Don't always accept the status quo, and don't just sort of use the easiest method, even if you know that it's probably not the best method. You got to think about what is it that you need at that point in time and go after it. That's what we did. So that's one. The second is find really bright, nice students to work with. Harry, he's a very bright young man, works hard, and we have a lot of fun. We've had enormous fun with this project. This is
Absolutely.
his very, very first publication that he's ever published. And I keep saying, to him,
Congratulations.
you're going to remember
It's a cool
this
one.
forever.
Yeah.
I think we should probably thank the entire sort of lab that's contributed to this. It wasn't just David and I, it was, a lot of people that took time out of their busy schedules to help with experiments and to interpret data and to sort of brainstorm this. It didn't work the first time either. It went through many iterations. And I think without those people, it wouldn't have ended up nearly as simple and elegant as it has.
So before we close, I have one more question. And that is, how long did you kick around this idea in your head before you started playing around with the dual tracers and really piloting the idea?
Yeah, that's a great question. It's funny. And it's true, I think, for a lot of us in science, right? Other people in the lab had tried this same method and failed. Okay. And, again, it says a lot about, moving forward in science. You can't just assume that because one person tried something that it's impossible. There are technical reasons for that. And so when Harry came along, he was particularly courageous, and we thought, let's give it another crack. Now, we did make some modifications, so he changed this, changed that, and bang, we perfected it. And I think that's such an important moral, that, and you hear this story a lot. This has been kicking around for 5 years at least, the idea.
Well, very cool. I want to thank you, David and Harry, for coming on the podcast, for sharing your expertise, and for really discussing this novel methodology to assess glucose uptake. I encourage our listeners to read the full article, as all of our featured articles are freely accessible online and are in the March issue of Diabetes. So thanks again, guys. It's a pleasure to have you.
Thank you.
Another paper we'd like to highlight from the March 2024 issue of Diabetes is titled Protein Tyrosine Phosphatase N2, or PTPN2, Regulates Metabolic Flux to Affect Beta Cell Susceptibility to Inflammatory Stress. We are joined by the senior author of this paper, Dr. Lori Sussel. Dr. Sussel is Research Director of the Barbara Davis Center at the University of Colorado Anschutz Medical Campus. Welcome, Dr. Sussel, and thank you for being a part of this podcast.
Thank you for having me.
All right, Lori. So we like to start with like a nice icebreaker question, which we didn't provide to you. So if you were not in science, is there a career do you think you would have pursued rather than being a scientist?
Yes, I've always said that if I didn't make it as a scientist, I would become a ski instructor.
Oh, that's a great answer.
Nice.
Yeah.
Downhill?
Downhill. I grew up in British Columbia, Canada, and I started skiing when I was 4. And it is one of my passions, although I don't get to do it as often as I'd like anymore.
Awesome. All right, I have the first question. So I would love for you to briefly summarize your research interest, your research program, and then how those interests led to this project.
Yeah, so I've always been really interested in the regulation of the pancreatic beta cells, and how they form during development was where I started, but have moved more into how they become dysfunctional in diabetes, both in type 1 diabetes and in type 2 diabetes. And in particular, in type 1 diabetes, although we realize it's an autoimmune disease and the main culprit for that is a dysfunctional immune system, it's really remarkable that the only target in this autoimmune disease is the pancreatic beta cell. So I am just perplexed as how that's happening and why it is the beta cells that are targeted and what happens to them during the autoimmune destruction, like how they contribute to the disease.
Yeah, that's really interesting.
So, Lori, can you tell us more about PTPN2 and why you're interested in it as a target for type 1?
Yeah, PTPN2 is a very interesting tyrosine phosphatase. So also its other name is the T cell protein tyrosine phosphatase. So a lot of people just associated it with the T cells. But interestingly, in type 1 diabetes, it is mutations in PTPN2 are risk alleles for type 1 diabetes. However, if you take a mouse model that knocks out PTPN2, the phosphatase, throughout the mouse, they get rampant autoimmune disease. However, if you just take it out in the T cells, you get a much milder form of the disease. And so our thoughts were that this phosphatase, which is also expressed in the pancreatic beta cells, were somehow compromising beta cell function in addition to making the T cells dysfunctional. In fact, it's also in B cells, so it makes both T cells and B cells dysfunctional. Our hypothesis was that the beta cells were also dysfunctional, and that was like sort of the trifecta, having the B cells and the T cells being prone to autoimmunity, and the beta cells being the target, also having these defects.
Sure. No, that makes sense.
Lori, in this paper, you generated a PTPN2 beta cell knockout, but in your initial glucose phenotyping experiments, the phenotype was pretty minimal. However, when you perturbed the system with inflammatory stimuli, and this I think ties in pretty nicely to what you just mentioned, now you start to reveal some important aspects of this gene and beta cell function. So, can you tell us a little bit more about those experiments and their key findings?
Yeah, so I think a lot of the field in diabetes or type 1 diabetes right now are realizing that there is, again, like I said, a sort of a joint problem. There's a problem with the beta cells themselves that make them maybe more susceptible to an autoimmune attack, and then a problem with the T cells. And so we weren't really surprised that the beta cells with the knockout of PTPN2 had only a very mild phenotype. Other people have published this using cell lines that you really had to, in models of type 2 diabetes, really stress the cells that had mutations in PTPN2. And once these cells were stressed, they actually succumb more readily to the stressful conditions and became dysfunctional. So we weren't surprised. These mice were pretty normal with the PTPN2 knockout in the beta cells. But then when we started to applying different stresses, more that the type 1 diabetes-like stresses, so treating them with streptozotocin or treating them with inflammatory cytokines, then they were much more susceptible to those stresses when they lacked PTPN2. So we felt like there was something in the cell that was making them, they weren't dysfunctional enough to become dysfunctional under basal conditions, but they were sort of on the brink of being dysfunctional when they had to face any stresses like an autoimmune attack.
Yeah. As a follow-up, you administered cytokines, I believe, systemically. What about something more general like LPS, which kind of, I know there's been links to intestinal permeability in type 1 diabetes.
That's a really good question. No, we didn't do that. We just basically used those two models, although now we've also crossed the mutation into the NOD background, which is where we're going in the future, because it just took us 10 back crosses and doing speak and just, it's taken us about 2 years. Plus it's a conditional knockout, so we have to cross in the pre-allele on the NOD background as well. So, those experiments I think will be the better, I have to confess, the better experiments than treating the islets with cytokines or treating the mice with streptozotocin. But yeah, LPS would have been another good one.
Interesting. Kirk?
So, Lori, I want to take a sort of a left-hand turn here. And there's some very interesting work on PTPN2 and the hypothalamus, right? And suggesting that maybe inhibiting it would help with leptin sensitivity. So the premise of that work by Dodd and colleagues suggested that, you may want to actually administer a therapeutic that inhibited PTPN2. Can you comment on your thoughts, how that might affect the beta cell? Because that's not something that they looked at.
Yeah, so I would be worried that, I think you could use it as a therapeutic target in the brain if it didn't actually affect systemically the T cells or the beta cells. I think that would be, you'd have to have localized targeting of PTPN2. And it probably also depends on the targets. Obviously, this is a tyrosine phosphatase. we started to identify some of the targets, but I'm sure there's a lot more out there. And the targets in the brain may be different than the targets in the beta cells. So it would really influence how you would develop those therapies.
Right. And I guess as a follow-up, conversely, what are the implications of maybe targeting the beta cell PTPN2 activation, right?
Yeah, so that's, I mean, again, if you activating PTPN2 also has, I mean, it could, or reactivating it, say, in the context of type 1 diabetes, could be a good thing, but also you don't want to activate it too much that it starts to have off-target effects. I think it is, like I said, a protein tyrosine phosphatase. It is an enzyme. So, I think we would need to understand more of the biology. What I thought was really interesting, what we didn't expect coming out from our studies, is the effect it had on a lot of the metabolic proteins that are affecting glycolysis. And so again, I think that beta cells, and probably a lot of the cells in the body, there's this fine tuning where you have to have just the right amount of metabolic output for them to be either resistant or not resistant to either an autoimmune attack or a high fat diet or any type of stresses. So it may be more of a rheostat in that sense. So again, too little is not good, but too much may not be good as well.
Sure. Thanks.
You foreshadowed this question a little bit, but what are the exciting critical follow-up studies that you either have on deck or even in an idealistic world of unlimited NIH funding that you would like to do?
The first thing is, yes, we finally have the PTPN2 knockout on the NOD mouse background. So the key is, does it accelerate diabetes in that background? And the female NOD mice get diabetes around 8 weeks of age. So we would see if it happened sooner, but the male NOD mice have much lower percentage of developing type 1 diabetes. And so we might actually see an increase in the diabetes initiation in male NOD mice. So that's really where we're at now. And the mice are aging as we speak. In the future, we'd really like to delve more into the metabolic aspects of what PTPN2 is regulating, what its direct targets are, and how it affects the metabolic output and sort of the metabolic fitness of the cells. I think that's one really novel aspect that we weren't expecting out of our studies.
Yeah. You mentioned that this gene might be, or phosphatase might be a rheostat. Do you think that it plays a role in type 2 diabetes as well?
Yeah, and these are not our studies. Decio Ezerek has done a number of studies, mostly in cell lines, but also looking at high-fat diet and how it affects cells that are deleted for PTPN2. And it has the same effect in that the cells are much more susceptible because they become much more dysfunctional when you treat them with either high fat or even expose them to lipids.
Interesting.
All right. So is there any take-home message from your work, this paper or otherwise, that you'd like to emphasize while you still have the floor here?
So I really need to acknowledge my postdoc, Dr. Young Kyung Kim, who is, unfortunately for me, gone back to a position in South Korea, where he's now going to be an assistant professor. But it was really his work that is in this paper. He did all of the experiments that were described in the paper. And he is the one that identified the metabolic defects. So he is the driving force behind this study, and he will be carrying this on in his own lab in South Korea. Yeah, so I think type 1 diabetes we've been studying its causes for a long time. And while I agree that the immune system is the major culprit in disease, I think we have to consider the target organ and why it is the target organ, or in this case, the target cell. There's two sides to this equation. So type 1 diabetes is an autoimmune disease, but we have to figure out not only how to tamp down that immune system, but how to protect the beta cells or actually make them more functional in the face of an autoimmune attack.
Very interesting. Thank you very much, Dr. Sussel, for sharing your expertise and discussing your recent work highlighting the role of PTPN2 on beta cell responses to inflammation. I encourage the listeners to read the full article in the March issue of Diabetes. Thanks to all of you for joining us on the DiabetesBio podcast. We hope you're enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature article we discuss each month is freely accessible at diabetesjournals.org/diabetes, and this is also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, or subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you next month.